Okay, we'll go ahead and get started. So where'd everybody go? There's going to be a lot of empty seats here. One exam scares them off. Okay, we had last few students come on in. So a couple of things. One, overall on the exam, compared to other classes, you did quite well. Don't become overly confident, but for instance, the average on the exam was about 4 points higher than last year's class. Um, as you saw, as I had them posted, some students are doing very well. There were 18 students who had 99 and above on the exam. I think 60 are in solid A's, and 115 or so are in B. And that score that I put on there does not include the clicker points. So those numbers are actually going to go up a little bit. If you didn't do as well as you'd hoped on the on this exam, change your strategy. Everybody familiar with Einstein's definition of insanity? All right, don't keep doing the same thing over and over and expecting a different outcome. So there will be a significant group of students who will change their strategy and how they prepare for the second exam. They'll come in and do markedly better on the second exam, and then the third and the fourth. Then that optional final exam at the end will then fully replace the first exam grade, and it disappears. Yes. Right. So the optional final exam is a cumulative exam. It is optional. When I post the grades after exam 4, they will include all the iClicker extra credit. They will be the grades that I would submit if you do not take the final exam. You want to be in the position where you can look at your grades after exam 4 and go, I'm good with that. I'm going to spend my time preparing for my other exams. Now, if I remember correctly, our scheduled final exam period is almost at the end of the exam period. So there's almost 10 days, almost approaching 2 weeks from the last exam until the final. Now, this is the way to think about that final exam. And if you think at this point you're going to be taking the final, I'd be preparing for it in the sense that obviously, I don't think you're going to have time to reread the entire book in preparation for the final exam. The exam will be a little bit longer, maybe about 60 questions, because they give us about a 2-hour time period for the exam. And we're going to cover 20, 22 chapters, something like that. So in general, I'm only going to have about 3 questions per chapter. So my goal is to think about what are the 3 most important points out of that chapter. So I would be reviewing as we go along, going back and reviewing that material, even just rereading the PowerPoint slides and that in preparation for it. Otherwise, if you take the final exam, I will essentially have 5 grades for you. I will simply average the 4 highest grades. Probably, generally, about 50% of the students who take the final exam improve their grade to some extent. Some, I've seen it change by an entire letter grade where they did very poor on one exam. Or it doesn't have to be the first exam. For some reason, they got 3 good grades, but for one exam, something personal happened, or there was some concepts that they just didn't have time to master, something like that. There is no jeopardy in taking the final exam. If it's the lowest of your grades, then it just gets omitted. Okay? So you can review your exams during my office hours until September 24th. I have to put these time limits on here or things get crazy, especially towards the end, and then I have a whole parade of students who want to look at all 4 exams. And it's just, with 300 plus students, it just doesn't work. Okay. The other is to remind you that the 4th exam is scheduled for the day before Thanksgiving. I didn't do that. The provost and the administration set the academic calendar. This is a regular lecture day, and it's the provost's policy that we meet on all days. And I'm about a half of a lecture behind. We're going to need those lecture periods. So I talked about this on the first day. You are adults now. So that exam will be there. If you choose not to take the exam because you want to travel or do whatever for personal reasons, you can use this final exam to replace a missed exam. Okay, or if you get stuck in traffic or whatever and you don't make it to class for an exam, that's not a justifiable reason, then you can use the final exam. Any questions? Okay. So what we're going to do is we're going to finish up chapter 7. This is a big chapter. It's an important chapter, and it's going to be a significant part on exam 2. Yes?
For the fourth exam, would it be possible, like, if we did have time to take a before that way?
Then I got to open that up to 300 plus students. So it becomes unworkable. Sorry, it just. Okay. If I made the calendar, I'd make Wednesday, Thursday, Friday the regular Thanksgiving break, but the people who make that decision are way above my pay grade. All right, so let's do the first 2 questions like we've been doing. So this is from the last one. This is a recombination experiment. You can use your calculators on this one. So again, the first 2 you do on your own. After that, you can collaborate. So you have a cross of 2 parents with these 2 genotypes. Is this a test cross? Yeah, okay. Not part of the question. There's the number of progeny that you get out from the cross. What is the recombination frequency between the genes A and B?
Yeah, Professor, you didn't change it to the numbers.
I'm sorry, what? Oh, sorry. I just want to see who was paying attention. Let's change this to. Come on. Let's try that. So the easiest is express it as a percentage, or you can do it as a decimal, 0 point something. Yes. I see a big B there, and I see a big B there.
In the pairs.
Well, who did that? Okay, never mind. You can't. How are you going to do it? So what would be the answer?
0.
Well, you can't. That can't be an outcome. How did that get on there? Okay. All right, so let's look at this question. Again, my typing skills. How can you have progeny that have a big B allele when there's no big B allele present in the parents? It's a very simple, unsolvable problem. Okay. Huh? I didn't do that on purpose. No. So I'll give you the credit for that one. That was my problem. Okay. So let's see if I can do any better on the second one. Okay. The answer would have been 23% if I had typed it correctly. All right, this is from today. So now we'll go back to. There we go. Come on. This one seems to be going a little better. We'll go to 1 minute on this one. While I'm doing this too, also, I looked, there's 256 registered iClickers, which looks like it's pretty close to the total amount, but at the end, I'll run up the registration again, but the in-class registration looks like a pretty effective way of getting them registered. My guess is there's only 15 or 20 missing at this point. Okay, there we go. So what's the answer? Everybody went pretty fast on it. Pure. Okay, so we're going to talk about this a little bit later in interference, it's really in something we call double crossovers. You typically observe fewer than you'd actually expect to see because of this phenomenon. So we'll get into more detail in a few minutes. What? Go back to. So the expected number of progeny is going to be fewer than the actual number.
Wouldn't it be greater?
Son of a bitch.
Because the number of games.
So this is going to be a really good day. All right. Maybe I'll just give up on iClick. Yes. Interference is the actual number of double crossovers, and we'll tell you what that is in a little bit later. The actual number is less than what you would expect because of the phenomenon of interference. So I got that one wrong too. Yeah, I'll give you credit for that. I think I did better on the exam. I don't think I found any that made no sense at all. Okay, so here's where we were. The key to understanding all these recombination problems, and we're going to get into the most complicated case, is going to be the so-called three-point cross. I'm going to walk you through it step by step, kind of like in calculus, you go through all the theory, and then they show you the shortcuts. All right? Will this be on the exam? Yes. This is the kind of problem that tends to take up 3 or 4 questions. So you're either going to get 3 or 4 questions right, or you're going to get 3 or 4 questions wrong. Because you got it. The important thing is to start with the double heterozygote and figuring out correctly whether the alleles are in the cis or the trans configuration. So we're going to do these with flies. These are Drosophila cross. So I'm going to refer to, I got a slide that I'll tell you in a minute, but I'm going to just refer to this. This will be the female, and this will be the male. Because in Drosophila, there's a quirk in that genetic recombination, intrachromosomal recombination, doesn't occur in males. So we're going to use the males for the test cross. The females will be the double heterozygote. All right. In these two crosses, the important point here is it doesn't matter whether the alleles start in cis or trans, you're going to get the same outcome. And even though we may be talking about 3 or 4 genes, I'll give you one example with 4, when we talk about are these genes in cis or trans, we can only really talk about 2 at a time. All right, so we got the 2. Is there any difference in the phenotype of these 2 females? What's the difference? Yes? One's green. Any difference between this? No, this is the male. So this female, this is a double heterozygote female, and this one. Is there any difference? So the female has got the same phenotype because she's just a heterozygote for the 2 genes, so she's got the 2 dominant phenotypes. Here, the alleles are in cis, I think. Yeah, in cis, and over here they're in trans. So the point of this slide is to show you get the same outcome. So this is the test cross. So there's homozygous recessive, so there's only going to be one gamete that comes from the male, okay, the 2 recessive alleles. And you get 4 different gametes that are possible with the female. The non-recombinant will be one that will have the 2 wild-type alleles, and one will have the 2 recessive alleles. And that's just simply this upper chromosome. These are the 2 homologous chromosomes. These 2 chromosomes with no crossing over simply give you these 2 non-recombinant gametes. Okay. Now, if these 2 genes were so close together that recombination never occurred between them, would you get these gametes? No, you just have the 2 parental or non-recombinant type. But then you've got the recombinants, and if you look, all that's happened is since these were in cis, recombination put them in trans, and so now you got the 2 recombinant gametes. How often that happens is dependent on how far apart those genes are, and that's going to be the recombination frequency. So when you look at when you make these crosses, you have 4 different phenotypes. Okay, again, this lower chromosome right here came from the male, so we don't care about that one. The first thing you want to do is look at the numbers of progeny in each phenotype. They should occur at almost equal numbers. So we have, this is a little too good. So you got 40 and 40 and 10 and 10. So these two obviously go together, and the most abundant is going to be the non-recombinant. It'll always be that way. So you got 40 and 40, so 80% are non-recombinant. And then you got 10 and 10, so 20% are the recombinants. So what's the recombination frequency? 20%. 20 out of 100, 20%. The important point of this slide is you come over here, now this female has the same phenotype, but the two genes are in trans. All right. So watch what happens to the phenotypes down here. Same situation. You got the non-recombinant gametes, the recombinant gametes. This lower chromosome comes from the male. All right. The non-recombinant progeny, are they the same as over here? No, because she was in trans, so now you got one wild type allele and one recessive allele on each one. But they still make up 80%. That's still the parental. And what's your recombination frequency? Just 20%. It's just that these recombinant gametes are now the same as these non-recombinants. So crossing over, just if it starts in cis, recombination just flips it into trans. If it starts in trans, it flips to cis. That's all it does. Okay? Yes?
So the non-recombinant has,
On which side?
on the right-hand side has the, it has the wild type, that he's not of the
Right. So this chromosome and this chromosome are these two chromosomes unchanged. So these non-recombinants, this upper chromosome, are the two homologous chromosomes exactly as they are in the mother. The lower chromosome, in all these cases, is coming from the father. Okay. So to just quickly review, you really just have three possible gene arrangements. All right. We can have genes on different chromosomes, independent assortment. This is what Mendel saw. So we can get recombinant gametes, but these are genes on different chromosomes. That's not what we're talking about in this chapter. Or you can have two genes on the same chromosome that are so close together that recombination essentially never happens. So they are always inherited together. This is where you'd only get two gametes, both parental or non-recombinant gametes. You would have no recombinant gametes. Okay? Or the more common situation, the genes are on the same chromosome, but they're far enough apart that some degree of recombination happens. So to give some terminology so I can write exam questions, interchromosomal recombination between chromosomes, this is Mendelian segregation, independent assortment. Okay? The intrachromosomal recombination is this crossing over in prophase of meiosis 1, which is what we're looking at in this chapter. All right. Now, one little hint, because initially you're not going to know whether these genes are on the same chromosome or not, is look at the recombination frequencies. If it's interchromosomal, again, if the genes are on different chromosomes, then the recombination frequency will always be 50%. If the recombination frequency is significantly below 50%, 20%, 30%, then you're talking about genes that are on the same chromosome. A little bit later, we'll talk about what happens when that recombination frequency gets up to about 45% or so. Are we really talking about different chromosomes or not? Okay, but we'll deal with. But for now, let's just say the recombination frequency is significantly below 50%, so we're dealing with two genes on the same chromosome. Okay. Before we get back to the mapping, just a slight digression, and I don't want to belabor this But this was an experiment that Harriet Crichton and Barbara McClintock did that really proved that this genetic recombination is in fact a physical exchange between the chromosomes. They gave them visual proof. So Barbara McClintock, probably arguably one of the best maize or corn geneticists and cytologists. That is, she could look at the chromosomes from the corn under the microscope, and she knew every little nuance of those chromosomes. Well, she found a strain of corn that just through some chromosomal aberration, something similar to what we're going to talk about in the next chapter, had a little lump of densely staining material, had a little sort of glop of chromatin on the end of chromosome 9. And it was an anomaly. It's not normally supposed to be there. She had another strain, which also on the other homologue had another little piece. So what happened was, fortuitously, this gave her some microscopic evidence that she could distinguish one homologue from the other. So that physically, she can determine the two of them. She could distinguish the two. So she looked at two linked genes that are on that chromosome, one for the coloration of the endosperm, and the other for whether they're starchy kernels or whatever. Okay. I'm going to let you work through this. She set up this cross. This will be a good test if you want to understand recombination. And the idea is to look for progeny that have the two recessive traits, this genotype. You should be able to convince yourself that the only way you can get that, the only way you can get this progeny from this cross is by a crossover between the C and the WX loci. When she did that, she also showed now microscopically that chromosome 9 had both of these aberrations on it. So it was a physical exchange between the two homologs. All right. So just provided that conclusive scientific evidence that during recombination, this chromatin, this DNA is physically being exchanged between the two homologs. Okay. All right. So back to the recombination that we've been looking at. So the arrangement of these alleles in a double heterozygote, as we just saw on that previous slide, will let you then predict the phenotypes of the progeny. Since you're using a test cross male, then you can look at the phenotype of the progeny and determine what the genotype of those progeny are. The recombinant phenotype, as we just did, determines the recombination frequency. All right. So here's another example. We'll try this. This is obviously not a fruit fly. All right. Let me see if I got this one right. So you do this cross, two genes, the T and the D loci. So here's the parent. Here's one. Here's the test cross. Are these in cis or trans? Okay. These are in cis, so recombination will put them in trans. Yeah, you can do it either way. I see all the different. You can either be 0 point something or a percentage. Yeah. It shows me all the different entries. I'll go to 2 minutes on this one. Okay. So what's the percentage? Okay. So you just have 16% recombinant gametes. Add those together out of a total of 100%, so it's 16%. All right. That's all there is. That's all there is to it. The ones on the right. Here? That's from the test cross. So here's the 4 gametes you get from the double heterozygote. Here's the non-recombinants. All you're looking at is how often does this happen? Again, all you're doing is saying, okay, in this experiment, what percent of the meiotic cells underwent recombination between these 2 genes? What percent? If it's 16% recombination, this is that question that about 50% of you are going to miss. We did this before. So it'd be 32%. Remember, only 2 of the 4 chromatids are involved in the recombination. So what this is telling you is in 32% of the meiotic cells, there was a crossover between these 2 genes, and that gave you 16% recombinant gametes. Okay. All that you come from this question is, well, the point I made before is if your recombination frequency is approaching 50%, let's say 45%. Well, does that mean that those genes really are on the same chromosome, but they're very far apart, or they're on different chromosomes? And there's some statistical tests and such that you can use. The chi-square is a little bit of an old one. We're not going to do any of that because the 3-point cross is where we're going with that. Can actually answer that question for you. So just to be aware that it gets a little bit, if your recombination frequencies are 25, 30%, yeah, you're dealing with 2 genes on the same chromosome. As you get closer to that 50%, it becomes, you have to have some additional data, and that's what we're going to do now. Okay. So here's the point. A so-called 3-point cross just means we're going to have 3 genes, A, B, C. Keep in mind that recombination can occur at any point between the 2 genes, and you still get the same outcome. All right. The whole mapping that we're doing with this is just based on the idea the further apart the genes, the more likely recombination is going to occur. And so the higher the recombination frequency, the further apart on the chromosome the 2 genes are. All right. Also that these distances are additive. So some of the nomenclature that's used, a map unit is equal to 1% recombination. So in that previous question, the 2 genes were 16 map units apart, or what's often very commonly used as a centimorgan. So this MU designation, the CM, or 1% recombination, they all mean the same thing. Okay. And also, if you kind of notice this additive nature, so the distance between A and B is 5 centimorgans, B and C is 10, and the recombination between A and C would be 15 centimorgans. Okay. Also, keeping in mind that, okay, that's what we just said, that your maximum recombination is going to be 50%. Okay, so before we really get into the mapping part of it, let's try this question. You just have 3 genes. These are the distances. It's 22 map units between R and S, 8 between S and T, 14 between R and T. What's the order of the genes? Again? Oh, God. All right. Try it again.
It's close.
There you go. Okay. So it should only take a minute for this one. All right. So what's the answer? Maybe. Come on. One of these. All right. All right. So we got that figured out. All right. Okay. Since that's a little too simple, we have to make this a little more complicated. All right. This is a so-called double crossover. The important point here is you have two crossover events between these two genes, between A and B. What that's going to do is put the alleles back into the original configuration. They're going to look like non-recombinants. Did recombination happen? Yes. You just don't detect it in your experiment. So the recombination frequency is based on all of the recombination events. So double crossovers, will that tend to, since you don't know about them, will that tend to overestimate or underestimate the distance between these two genes? It's going to underestimate it. Okay? So what happens here is we have the big A, it crosses down here to the blue chromosome, but then crosses back up to the red homolog and back out again. So we've had two recombination events. You can see that here the DNA between the genes has been exchanged between the homologs, but your experiment is tracking the alleles and they look like they're in the original parental configuration. Okay? So this is something we have to think about. So the further apart your genes are, the more likely this double crossover is going to influence your experiment. And initially, you can't really tell to what extent. Empirically, it's been observed that this phenomenon of interference that we'll talk about, when you get to about 10 map units, then the probability of the double crossover is nearly zero at that point. Okay. So we just started out with an experiment. We're looking at some test cross. We're looking at 4 different genes, A, B, C, and D. So each other paired up. So A, when the recombination frequency between A and each of the other 3 genes is 50%. So what does that tell us? Most likely. That A is probably on one chromosome and the other 3 genes are probably on another chromosome. Okay? Probably. B and C is about 20%. Are these going to be on the same chromosome? Yeah. The 10% here. These 3, these are really telling you before we even look closely at it, these 3 genes are probably on the same chromosome. A is on another chromosome. Okay. So a term I've used before, we call these linkage groups. They're the equivalent of chromosomes. It was just before people really understood what was going on. If the genes are linked together, because the recombination frequencies are well below 50%, they just said, oh, they're in the same linkage group. In a physical sense, it's because they're on the same chromosome. Okay, so we can start to diagram this out. So we'll put A in linkage group 1. It could be chromosome 1. Do any of these experiments tell us what chromosome we're dealing with? No. Later on, we got to talk about how we can deal with that. Then on linkage group 2, we'll just call it arbitrarily chromosome 2, we have 20 centimorgans between B and C. Now, do we know whether B is down here or in the middle? No. All this is telling us is the linkages, the relative distances between them. It doesn't tell us anything about whether they're close to the telomere, whether they're close to a centromere, what chromosome they're located. We have to use other tools to find that. Okay. So the tricky one is, so we've dealt with A, B, and C. The question is, where does D go? So does D go over here to the left of B, between the B and C, or to the right of C? Okay. Well, if D is 10 centimorgans to the left of B, then the distance between D and C will be 30 centimorgans. Right now, we'll just ignore the double crossovers. If it lies to the right of B, the distance from D to C would be about 10 centimorgans. When you look at your actual data, it's about 28%. So that's close to the 30. So this is the best fit of the data. So this is the organization of the genes along the chromosome. Just taking advantage of recombination frequencies and that these recombination frequencies are additive. Okay, any questions on that? Okay. So this is the so-called three-point cross. So I'll walk you through this. This is probably the most complicated situation we're going to deal with. And it's also very efficient at helping you put these three genes in the proper order on the chromosomes. Okay, so there you go. Any questions? All right. This very busy, colorful slide really just shows you a couple of things. Here's our four chromatids, the red homolog, the blue homolog, three genes, A, B, C. Are each of these pairs of genes in cis or trans? If A and B are in cis or trans? They're in cis. In this configuration, any pairwise combination, they're all in cis. So a recombination will simply put them in trans. Okay. We have three possibilities. We got a single crossover between A and B, a single crossover between B and C, or the double crossover, but the important point is it's going to occur on each side of B. If it occurs here and here, we will detect it in our experiment. What if it occurs between A and B? You're not going to observe it. But if we get a double cross, so all the double crossovers we're talking here is essentially both of these things happening. Okay? So these are the only three possibilities that we have. All right. So let's see if there's a chance that I actually did this question correctly. Let's see. Before we get into the actual mapping. Looking at some of these crosses is a little bit like looking at stereoisomers in organic chemistry, making them rotate.
No, chem is so much easier than this.
It is?
Yeah.
I don't think so.
This is compared to
What?
the 1 and 2 are those compared to the
Oh, really? I don't know. When I was an undergrad, I had a lot of friends who were pre-med. The two classes they had the most trouble with was physics and genetics. But those are my favorite ones.
genetics is hard.
Let's see. All right. Okay. I think it looked like. So which one can't happen?
C.
There you go. It's easy. Nothing to it. Why couldn't that happen? Well, first you'd have to, the wild type alleles would have to be coming from the parent, from the other parent. Okay, good. All right, so now let's get a little bit, okay, so this is the 3-point cross. I'm going to walk you through this. Some of you are going to see it and go, oh, I got it. Others, it's going to go, what? You need to do a couple of these problems, I think, to figure it out. I think I have a couple of extra problems that are in the on Blackboard. They're from other textbooks. They're just more problems. I think you do a couple of them, you begin to see there's a pattern here, and they're really not that difficult. Okay, so as I've been saying, we don't have recombination in Drosophila in males. That's just kind of a genetic quirk. So in all these experiments, the test cross is going to be the male. The triple heterozygote will be the female. So we're going to start all of these in the same way. A triple heterozygote for the female and a test cross male. All right, so this is a simple situation. Every pairwise combination is in the cis configuration. All right, very similar to what we just had. All right, the way you want to break this down, first look at the progeny. There's going to be 8 different phenotypes based on 8 different genotypes. The first thing you want to do is look at the numbers and pair them up. So that's going to put you into 4 pairs. The most abundant group is going to be the parental, the non-recombinant. That's going to tell you the configuration of the alleles in the mother. And the double crossover, the rarest group will be the double crossover. That's going to set the gene order. Once you do that, the problem is pretty straightforward. If you don't do those steps correctly, you probably have everything wrong. Okay, so we're going to have these 4 pairs. As I said, the most abundant will be the parental or the non-recombinant. The least abundant, the double crossovers. And the 2 intermediates are going to be just the 2 crossovers that flank the center gene. Okay, so we did the cross. Here's the phenotype of the progeny. Most likely, I would just give you the genotype because it's just easier. All right, could figure out from the phenotype what the genotype is, right? Okay. Again, this bottom chromosome in each one of these 8 crosses came from where? From the father. So we're not going to worry about that one. So we're really looking at the chromosomes that are at the top of each of these pairs. So first thing we did, we look at the groupings. We got 283, 278, so these obviously go together. 50 and 52, 43 and 41, and 5 and 3. So we got 4 pairs. This is going to be the non-recombinant. So this chromosome and this chromosome are the 2 homologs in the mother unrecombined. So that's the original configuration of the alleles. So she's got all 3 wild-type alleles on one homolog and all 3 recessive on the other. Now, what you want to do first is determine the gene order. You only have 3 genes. Really, you're just determining which one goes in the middle. Does it matter which one's on the left or right? That's just arbitrary. So the important point is get the right gene in the middle. Okay? Don't fall into the trap that just because I give you the 3 genes that that's the order they're in. I'm not going to make it that easy. Okay? It just happens to be the way it is in this diagram. I think a couple of those problems that I put up on Blackboard, the genes aren't in the right order. So go to the double crossover. And I think the next one, okay. What you want to compare now is compare the parentals. So here's the abundant group. So these are the non-recombinant. So we have all 3 wild-type alleles. We have all 3 recessive alleles. Look in the double crossover and see which allele is flipped. Which alleles are different. Which one's different here? The SS allele, right? That's the one that's in the middle. That's the one that got changed by the double crossover. So compare the double crossover, the rare group, to the parental, the most abundant group, and that will tell you what gene is in the middle. The other two groups just have to be the single crossovers between the gene in the middle and one of the flanking genes. So what you want to do now is you got to be sure, okay, so now we got this 50 and 52. Are we talking about here or here? Again, watch the genes. Up here, ST and SS are in cis, right? And what about down here? Are they in cis or trans? They're in trans. You got ST, SS plus, SS, ST plus. How do you know it's not the recombination between the SS and the ebony allele? Because they're still in cis. They haven't changed. It's the one that's changed. So now you're looking at the recombination between ST and SS. All right. So all you're doing is adding them all up. There's 755 progeny, 102. For that single crossover is 13.5%. And here's the last step, and it's the most likely one where you'll get caught up. Remember, recombination frequencies are based on the total number of recombinations. So you also had a recombination that occurred down here in the double crossover. So you have to add the double crossover distance to the single crossover. So that's why on our map, this is 14.6 because it's 13.5 plus 1.1. Okay, you had 8 double recombinants, so you had a 1.1% double crossovers. The other side, 84, which is 11.1, add the 1.1, you got 12.2, and you're done.
So you add each intermediate with the...
You take each single crossover and add the double crossover because it happened on both sides. Like I showed at the beginning, the double crossover is really one of each of those single crossovers. So that's why you have to add it to both of your single crossovers, and you got the map. Okay?
Can you explain why you're giving 14.6?
It's because of the double crossovers. So you have 8 double crossovers, or 1.1%. Since that double crossover actually is 2 recombinations, you add that to the single crossover difference.
And that is what you would want us to find for the exam.
Yes. And a problem like that ends up being 3 or 4 questions. And so it's either 3 or 4 you get right or 3 or 4 you get wrong. But I think if you just do a couple of those, you see they just follow the same pattern. Yes. Why did I separate? Okay.
And then you also took the percentage for the combination of the right.
Right, because this is a single crossover between this gene and this gene. But if you look here, these 2 alleles, SS and E, are still in the cis configuration, which is exactly the way they are in the mother. So there was no recombination to the right of SS. So this tells you this is the recombination between SS and ST. This one is just simply the recombination on the other side. So one's a recombination for over here, and the other is a recombination for over here. Is there another question? Yeah.
Yeah, I was going to say, are you willing to show us how you got the middle recombination again?
No.
Okay.
I mean, what, you mean the double crossover? This one?
Yeah.
In other words, this one's key. You have, if you don't get the right gene in the middle, everything's wrong. Nothing will make any sense at all. All you're doing is saying, okay, take this upper chromosome here, and this upper chromosome here, because the lower one is coming from the test cross male. here is, we got 3 wild type alleles on this chromosome and 3 recessive alleles here. Now go to the rarest group, the smallest group, that'll be the double crossover. Compare those, compare these and this to up here, and what's different? See? This one's got the wild type allele here and the recessive allele up there. So the SS allele is reversed compared to the parent. The one that's different in the double crossover is the gene that's in the middle. Is that the question?
Yeah.
Okay. Like I said, if you do a couple of these, I think you'll see how it works. Yes.
So how do we get percentages?
Percentages?
Yeah.
Oh, it's just the total number here, down here, divided by the total number of progeny. So you got 755 progeny, and of those, 102 are in this single crossover, so 13.5%, 102 divided by 755. So you just take each of these numbers divided by the total. Okay. So it's described in the textbook. I guess I think there's an animation on the resource page that the publisher has. I've put a couple more problems in there, so I think it's just one of those. If you sit down and with your beverage of choice and kind of work your way through it, you should be able to see how it works. And it was through those kind of experiments that gave you this genomic map for, this is the one for Drosophila. Is this probably like most of the genes in Drosophila? There's about 13,000 genes in Drosophila, so there's only a few of them. So we only have about twice as many genes as a fruit fly. Question is, how does that work? Okay. So that brings us to this question that was at the beginning of the section, is interference. And that is, in most cases, when you look at the actual number of double crossovers, it's less than what you would expect based on the probability. All right. So in this experiment we just did, so this is exactly the same map. I just took this off of the other figure. What would be the probability? What's the probability of, or how many double crossover progeny would we expect? Okay. So if we again treat these as independent events, that these are really 2 independent crossovers. So if they're independent events and they don't influence each other, then we simply multiply their probabilities. So we have 0.146 times 0.122, 0.0178 times the 755. We would expect about 13.4 flies. I don't know what a 0.4 fly looks like, but we would expect 13.4. But how many did we actually see? If you go back to 8, if you go back to the experiment, we actually saw 8. So we expected 13, we only saw 8. This is this phenomenon known as interference. The rationale behind it is the presence of the crossover. These DNA strands are actually physically crossing over with each other. The presence of the first crossover interferes with and prevents the second one from happening. So does it make sense that if the genes are further apart, it's more likely that that crossover will happen? You'll have less interference, and you'll get closer to what you expected to see. And then as the genes get closer, that interference increases. And as I said, we kind of empirically determined that once you get to about 10 map units, or 10 centimorgans, then interference is essentially 100%, and you see no double crossovers between the 2 genes. All right, but we have to make it confusing. So we have the coefficient of coincidence. That is simply the number of observed double crossovers, in our case 8, divided what we expected. So the coefficient of coincidence is 0.6, which really says we saw 60% of the expected number of double crossovers. The interference is just 1 minus that coefficient. So the interference is 0.4. So what happens to interference as the number of observed double crossovers goes up? The coefficient of coincidence gets larger, and the interference goes so the interference goes to, okay, so does that make sense? So let's say we had 13, we observed 13, we expected 13, the coefficient of coincidence would be 1, and the interference would be 0. There'd be no interference. We saw exactly the number we expected to see. Yes. Okay, so let's try this question and see if you... I can't imagine I actually got this one right, but we'll try it. So does this one have 2 correct answers again? Okay, I'll go to a minute and 45 seconds. Then. Okay, so what are the correct answers? A and C, okay. I didn't put it on there, but as I'm rereading it, okay. So so the coefficient of coincidence is telling you you're going to only see 41% of the expected number of crossovers. And the interference is saying you're not going to see 59%. They're really saying the same thing, just expressed differently. Okay. So trying to tie this back now using this kind of genetic mapping and recombination and tying it back to the pedigree analysis that we talked about in chapter 6. This was one of the ways that was used early on in trying to identify the genes that are associated with some specific human genetic disease. So, for instance, they did this with Huntington's disease. It took about 20 laboratories. At the time, the technology, this was probably from about 1980 to 1990. I think it was done in the 80s. It basically took almost a decade to find a DNA sequence that was close to the gene that they think is responsible for Huntington's disease. It took another decade to actually map out the gene. So it literally took 20 years. Now, with genomic mapping, that would be I don't know how fast it'd be done, but it'd be a whole lot less. Probably a day or so if you had to find a new one. But what the idea here is, can we in fact find a gene that seems to be responsible for a certain human genetic disorder, but link it to a marker, a gene that we can follow easily? So that's the example I'm going to give you here. So this nail patella syndrome It's a dominant human genetic disease, but it appears to be closely linked to the locus for the ABO blood typing. So it's a rare autosomal dominant condition. We're going to assume that every affected individual is a heterozygote for the reasons we talked about before, and that unaffected individuals are going to be homozygous small n. So in this case, the small n, is this the wild type allele? Yes, because this is a dominant mutation and using a capital N to indicate that allele. Okay, so here's this pedigree that we've seen before. If we just kind of work our way through it, so here's just a reminder that, so the blood type is shown in the circles or the squares. The filled in squares indicate affected individuals. So we're assuming every affected individual is going to be a heterozygote for this gene. And as you work your way through this pedigree, what you see is this big N allele is being co-inherited with the small I allele from generation to generation. Okay? And it follows all the way through except for these two situations. So why don't these two individuals actually have the disease? But why? What happened? Okay, so if we look at the parents, how do you get this genotype from parents 5 and 6? The small I, small n could come here, right? But how do you get another small I, small n allele? Because of recombination. Okay, so that's what, if the genes are close enough, they'll generally track together and tell the investigator that the N allele, the N gene is close to the locus responsible for the ABO blood type, but it's far enough away that occasionally you do get recombination, and so that's how you explain this individual and this individual. Okay, but it was a way of trying to follow and find a gene related to this human genetic disease by being able to track it with another gene that was nearby but much easier to assay for. Okay, this was all done before a lot of the molecular tools that we have now, so this is much less of an issue. All right, everybody see how that works? Okay. So getting a little more up to date of what we're doing now is really, this is the basis of DNA fingerprinting. This has been around for quite a while now in different variations. It originally started out with so-called restriction fragment length polymorphism. Does everybody know what restriction enzymes are? Finally, after 30 years, they actually teach you about this. Okay. So restriction enzymes are these bacterial enzymes that cut DNA at a very specific DNA sequence. And so they were used to analyze DNA, and depending on whether the restriction site was there or not, you get different length DNA fragments. I'm not going to go into the real details of this, because this is up here more of a historical note. It was very time intensive, laborious, pretty much hit and miss trying to find the right situation, finding these sites, but it was all that was available at the time. After that, with more analysis, came out so-called microsatellites. And that was the discovery that we have these short little DNA sequences that are repeated, tandemly repeated, that is, one after the other, a number of times in the genome. And so now you might have someone who has 9 copies and another individual with 4 and 3 and 2. So it gave us actually more alleles of that gene, literally, and it was easier to do and gave us more markers to sort of mark and put on the genome. Now we're at the highest level of analysis that we can do, the so-called single nucleotide polymorphism. Polymorphism just means variation. But with the ability to sequence entire genomes now, we can actually look at variations at the single nucleotide level. It's estimated that if I took take two of you who are unrelated in here, compare your DNA sequences, you'll probably be about 99.9% identical and vary at about 0.1%. But since your whole genome is about 4 billion that should be a superscript. Since your whole genome is about 4 billion nucleotides, that means that between any two of you, there's about 4 million nucleotide differences. And those have now been mapped around the genome as little molecular markers, little signposts to tell us different positions around the chromosomes and such. And we can use those now to track with genes to see which ones are closest to these so-called SNPs. Okay, so millions of these little SNPs have now been mapped to the genome. And as I said, you can compare those. So you basically get a genotype of the unique SNPs for that particular normal individual, and you can compare that to an individual with a specific disease and help you start to track down exactly where in this 4 billion nucleotides is the gene that you're interested in. Okay, and we'll get in later on. We'll get into the technology more of how you do this. The other is actual physical chromosome mapping. So far we've done these genetic maps, but we don't know which chromosome they're on. We don't know whether they're near the centromere, whether they're near the telomere, are they on the long arm, the short arm. Where are these individuals located? And except for X-linked genes, obviously we can track those because of the X and Y. But if they're on the autosomes, this genetic mapping doesn't help us and tell us which gene is involved. The other thing to keep in mind, I'll come back to a couple of times, is I've said that this recombination mapping, this 3-point cross that we just did, assumed that crossing over is random. But it's not. It generally is. But there are places in the genome where recombination, so-called hotspots, where recombination happens more frequently than you would expect by random chance, and other areas such as around the centromere where there's almost no recombination. So that does interfere with our gene mapping. Okay. So here's the 4 we're going to talk about and show you basically how this works. Deletion mapping, somatic cell hybridization, in situ hybridization, and DNA sequencing. Okay. Here's the idea with deletion mapping. Deletion mapping means there's some part of the chromosome missing. If the part missing contains the gene I'm interested in, then how can I detect that in a genetic experiment? This was actually how the gene responsible for Duchenne's muscular dystrophy was tracked down. It was known to have been an X-linked gene, but the question was where on the X chromosome was this gene and with its mutant alleles. But when you started looking carefully at the chromosomes from individuals who had the disease, they all had the same small little region of the X chromosome missing. So I told you in that little region of the chromosome that was missing, somewhere in there was the gene that you were interested in. And then that allowed the mapping, and eventually they mapped it down to the short arm of X chromosome and actually eventually moved in onto the exact gene itself. So here's how it works. You notice this chromosome is shorter than this one because this region in red has been deleted. Literally, the chromosome was broken, and the two ends were brought back together, and the piece in the middle was lost. All right, in the next chapter, we'll tell you about how that happens. Okay, so now you have a homozygous recessive individual, and will this one be wild type or mutant phenotype? It's wild type. It's a heterozygote. Its allele is actually the deletion. Okay. Now, if the gene of interest, if A, is in this region that's marked in red, it's going to be missing from this chromosome, right? So now when you do the crosses, 50% are going to be wild type because they're just going to be a regular heterozygote. But 50% are going to have the recessive allele and the deletion. Does this one have the wild type allele to basically mask it? No. So you see the mutant phenotype. All right. In the jargon, we say the deletion uncovers the allele. So this tells you that your gene of interest is in this deletion. Now over here, let's say the region of deletion that you're looking at is down here. Here's the red. So it's missing over here. What do you see in the progeny? They're all wild type because they're all heterozygotes because the A gene is not missing. So this kind of deletion mapping, and once you knew where that deletion was on the chromosome, now you could begin to map where your gene would go relative on that chromosome. Here we know that this is fairly close to one end of the chromosome. Okay, so this is deletion mapping. Any questions? Okay. The other one that's been really useful in mammalian genetics is somatic cell hybridization. This is where you do something completely unnatural and get an interesting outcome. So you take cells from two different species. We're going to do this with mice and humans. Now, so this is in vitro. You're doing this in the lab. So you take cell cultures and you fuse the cells together. And there's different ways of doing this. You can use polyethylene glycol, and there's some other ways of doing it. We won't go into the technical part of it, but you fuse the cells together, and you end up with a cell that has actually two nuclei temporarily. So we call that a heterokaryon. All right, so now I got another question for the exam. So you got these two nuclei, and then the nuclei begin to fuse together. So now you got this full somatic complement of human cells and this full complement of mouse cells. And after they fuse, these cells actually begin to divide. But as they divide, they start to lose chromosomes. And they lose them randomly. And it's not really known why this happens. But when you do this with mouse and human hybrids, it's the human chromosomes get lost and the mouse chromosomes get retained. So you're going to end up with different cell lines that are going to have mouse chromosomes, but different numbers of the human chromosomes. And eventually these cell lines stabilize and you can maintain them. So you literally, there are available whole banks of these mouse-human hybrid cells that each clone, each hybrid cell has different numbers of the human chromosomes, and sometimes they're broken pieces and such. So the way you can make this, so this is just to kind of show you real quick. So you're just fusing them together, making the heterokaryons and getting through here. So let's just make this simple. You got 3 cell lines, and your gene makes product X, and you're trying to figure out where that gene is in the human genome. Well, if you assay cell culture 1, you find out it's making product X. But it's not being made in line 2 or 3. So what's different about them? Well, would it be on chromosome 2? You know you're making it, so it's got to be on chromosomes 2, 4, or 11. But it's not making it here, so it can't. It has to be on chromosome what? 4. But you got a little bit lucky because this strain actually has a deletion and this part of the chromosome 4 is missing, and this cell doesn't make product X or whatever it was I said. So the gene that codes for that product has to be in this region of chromosome 4 that's present here, but missing here. So this is a very effective way of at least getting some rough mapping of the human genes onto the right chromosome. Okay. Another very widely used technique in situ hybridization, it oftentimes is called FISH for fluorescence in situ hybridization. And that's because you're going to be looking at a karyotype of the chromosomes. but they've been prepared in such a way that you can still see the morphology of the chromosomes. But for instance, there will be a DNA probe that will have homology to a specific gene on your chromosome of interest. And that probe is going to be labeled, is going to be fluorescently labeled. So that probe is literally the magnet that's going to help you find the needle in the haystack. So in this example, this is a probe that's now made for a specific DNA sequence that's on chromosome 9. So wherever we see the fluorescence tag, we know that's chromosome 9. So can't see this quite so well with the lights, but you got all these blue chromosomes. Why are these chromosomes blue? Sorry, what? Yeah, these are probably stained with a dye called DAPI, which basically just stains DNA. So it's just giving you the shapes of the chromosomes. But here's chromosome 9 and chromosome 9 over here. And so probes have been made against all 26 pairs of chromosomes. And so you can do some very specific work in this way. So this is a very current, up-to-date, and widely used technique. Okay. Make sense, or are you just kind of drifting away? Yeah. This is what's probably changing all of this. A couple of years from now, I may end up deleting all these slides. Okay. Just like there's a lot of technology that's, your textbook's very up to date, but it's got some technology in the technology chapter, wherever it is, 19 or whatever, that is like, why is it still here? Because nobody's doing this anymore. This will be part of a history of molecular genetics course or something. DNA sequencing is just changing all of this. It is just, the important point here, it has become so cheap and so efficient to do DNA sequencing that the good news, I used to teach all the chemistry behind DNA sequencing. I made you guys know what it was. Now all you have to know to sequence DNA is that you put it in a little plastic test tube and you take it to FedEx and you mail it, and 2 days later you get an email that says, here's the link, here's your data, and you get it. Okay. I've got thousands of dollars worth of sequencing equipment in my lab. I don't know what I'm ever going to do with it, give it to a museum or something like that. Actually, I have this little power supply. It's an old power supply. It was in the lab I inherited when I started here. It's got these things called vacuum tubes in it. We put a digital meter on it. Actually, it's pretty accurate. But there's a mock-up of an early biotech lab at the Smithsonian, and I was there looking at it and I went, sometimes I've got that same power supply. So the same power supply I have in my lab, as far as I can tell, it's the same model is actually in the Smithsonian. I guess that just says how long I've been doing this stuff. All right. So this is what's happening. So the sequencing, the fact that you can sequence an entire human genome for under $1,000 now, probably in a couple of days, is we're just getting so much of our data by just getting the straight DNA sequencing. So this is really the physical map, and that is the nucleotide by nucleotide sequence as you go from one end of the chromosome to the other. So what this is really allowing us to do is, if you really want to know what genes are involved in cardiac disease, and it's probably, who knows, could be 100 genes, could be 10, could be 1,000, who knows. You really need a nice sample of individuals who have cardiac disease and a group of people who don't have cardiac disease, and to be able to do a meta-analysis and compare all of those genomes. So what was started, I think this has been completed, it's actually been expanded out, was the so-called 1,000 Genome Project. And that was to just start sequencing huge numbers of genomes. If you find this kind of analysis really interesting, and a huge area now is bioinformatics. This is just in probably a 10-year period, 10 to 15-year period, it went from a very specialized, almost black art known to a limited number of people in the pharmaceutical industry to a tool that everybody is using now. So in terms of getting employed, bioinformatics is a real skill. You bring that it kind of moves you ahead of all the applicants who can't even spell it, sort of thing. Dr. Narayanan, who was at Roche Pharmaceutical before he came here to FAU, this is his expertise, this sort of genes to drug paradigms, looking for genes that for cancer targets, this sort of thing. He's teaching the undergraduate version of bioinformatics in spring. It's a very hands-on, lab-based computer course. No programming. It's a real users. He gets into the tools and such. There's so much information on these databases now, and it just keeps expanding. So, in the last 10 years, the human genome has become a really tractable model system where the tools are getting to be almost as good as they are for Drosophila and some of these other organisms. So I also think those of you who want to go into human health, just the areas of genetic counseling for everything from prenatal counseling to disease tendencies and such. I think this is going to be a huge area in the coming years. Okay. So this just kind of reemphasizes something I said earlier about the fact that we made these genetic maps based on genetic recombination under the assumption that crossing over is random. And I think one of the hallmarks of doing research in science is that assumption is the mother of all screw-ups. And so we make these assumptions, and what we found was when you did the physical map, when you actually sequence the DNA, okay, let's do it this way. Were the genes in the right order? If you did your experiments correctly, would the genes be in the right order? In most cases, unless they're really close. But what was different was the distance between the genes. Sometimes they were closer and sometimes they were further apart, depending on whether or not this recombination was occurring randomly. So as we just said earlier, if you have a site on the genome where recombination occurs more often than you would expect by random crossing over, is that going to make your gene distances appear shorter or further apart? If you're getting crossing over more often than you would expect, it's going to get you a larger number, and your genes are going to appear to be further apart than they really are. Now, if your genes are close to for instance, the centromeres, where there are molecular mechanisms in place to suppress crossing over, then are those genes going to appear to be closer or further apart than they really are? They're going to appear to be closer. So there is this compression and expansion as you compare the genetic map to the physical map. Which one would you trust? Your tenure, if you're just whether or not you're going to get tenure is based on this decision now. Which one are you going with? I'm going with the DNA sequencing. Okay. All right. So let's see what you can do with this. Did we cover everything there? Actually, I'll pedigree now. Okay. All right, you're getting this one pretty fast, so. All right, we'll just go to 1 minute on this one. All right, so what's the answer? How do I do it? Sometimes. Okay. So that's chapter 7. In the next, about 15 minutes, I'm going to give you an introduction to chapter 8. Conceptually, I think chapter 8 is a little easier. I think probably, in my opinion, Probably chapter 7 is going to be one of the more challenging chapters for exam 2. Unless you don't like chemistry, then it'll probably be some of the molecular work. So, okay. So any final questions on the recombination? Yes.
It was actually sequencing. When you put your sample in the test tube and you take it to FedEx, what happens to it?
Oh, okay. So what you do is you have to purify the DNA. So, for instance, one advantage of the polymerase chain reaction is it's a very fast way of generating a specific DNA sequence. And then you get enough DNA, you literally just put that in a test tube. DNA is really a very remarkable molecule. It's one reason they've been able to sequence ancient DNA. I think one of the first ones, again, going back to the Smithsonian, if you've been there, what is it, the Museum of Natural History, there's the big woolly mammoth out in the front. And they took a small sample of tissue off the back of this woolly mammoth and sequenced it. Because DNA is a very, very stable molecule. So you basically just ship it by FedEx. You send it to a company, and
What do they do?
basically all the instrumentation, the older school used a chemistry that Sanger won the Nobel Prize for, but now basically it's been done what's called next-gen sequencing, and it's just a very high throughput, fully automated system. And so the technicians literally, you can basically be a community college trained technician because you're just going to take a sample of this DNA and load it into the instrument. It's all fully automated. All the sequencing, all the reading. I mean, I did it old school, and you literally went down the gels and read all the bases. Now it just goes. It's all read completely into the computer. It's all based on intensities of the signals as they come out using something called capillary gel electrophoresis. So it's a very efficient. If we send them a sequence of about 1,000 base pairs, I think it's somewhere between $5 to $10 to have it sequenced. It's usually the FedEx bill is more than the. I got to send at least 10 samples to be even worth the FedEx bill. At least because DNA is stable enough, instead of shipping it on dry ice or on wet ice, we can ship it at ambient temperature as long as we do overnight shipping. So yeah, it's like oligonucleotides. So many of these techniques we use synthetic pieces of DNA. You make those by sitting down in front of the computer and going to the website and just typing in the sequence that you want. The same thing, they've gotten very cheap, about 50 cents a base. $1 a base will give you enough primer to do about 2,000 PCR reactions. So those reagents are really quite inexpensive now. Okay, so chromosome variation. Now we're going big scale. What we're looking at here are changes in the chromosomes large enough that we could actually see these differences under the microscope. So these are going to be big changes in the chromosome. They're going to involve more than one gene. They're going to involve more than 2 genes. They're very likely to involve very large numbers of genes. So we're really just looking at the morphology of these chromosomes and how they're changing, but it's on this scale where we can actually see these differences if we're using the microscope. So the way the chapter is broken down into 3 parts, we'll do chromosome rearrangement. We'll talk about, let's see, have we talked about one of these yet? That deletion mapping would be one of these chromosome rearrangements. Aneuploids, which is the gain or loss of a whole chromosome. Basically, except for the sex chromosomes, cells don't like this very much. Or polyploids, which mean gaining entire sets of chromosomes. And these tend to be, you find these most commonly in plants. Okay, so just to kind of get started on this chapter in the 10 minutes or so that we have, 4 types of chromosome rearrangements we're going to talk about. We're not going to cover all 4 in the next 10 minutes. I think they're pretty self-explanatory. Duplications, that is a segment of the DNA in the chromatin is getting duplicated. So it's going to actually gain genetic information. Deletions, that's just the reverse of that. That's what we just looked at, a loss. Okay. Inversions, we're going to keep it simple. There's not going to be any loss of DNA sequences, but the sequences are going to get flipped over. So they're going to change the orientation of the genes on the chromosome. And translocations, which we won't get into today, are actually exchanges of genetic information between non-homologous chromosomes, like chromosome 9 and chromosome 14 kind of thing. And under that, I'll show you one example of an inheritable form of Down syndrome due to something called a Robertsonian translocation. But in the next 10 minutes, we can cover some of the basic ones. Geneticists like to use the term tandem. Whenever they're talking about tandem, they mean next to. So a tandem duplication is a duplicated sequence right next to the original sequence. So if you just use these letters as arbitrary indicators of some positions along the chromosome, then the segment represented by E and F has been duplicated, and it's exactly next to it, so it's a tandem duplication. So we have lots of sequences in our genome, what's kind of now called the dark matter of the genome, that in some cases may have thousands of tandem duplications next to it. A displaced duplication is exactly the same thing, except there's some DNA between the original sequence and the duplicated sequence. So everybody see the difference just between this tandem and displaced? It's just the position of the duplication. Okay. You can also have a reverse duplication, which is really just a combination of an inversion and a tandem duplication. So this segment represented by E and F has been duplicated, but it's also been flipped over. All right. Would that affect gene expression? Well, it may or may not. We'll talk about later on why it may have no effect or it may have a profound effect. Okay. So this is just showing you in cartoon form what we just showed. So here's the duplicated region. Okay. So here's one example of a duplication in Drosophila. It's called the bar-eyed mutation. So I should have a pic. I don't know if I got. So Drosophila normally have this large, very oval-shaped eye, and there's a small segment of DNA on the X chromosome, and as it gets more and more duplications, this eye shape gets more and more extreme, so it almost becomes just a narrow slit. Okay, so this so-called bar-eye mutation. So the heterozygous females are going to have smaller eyes. I'll show you an example in a minute. Actually, it might. Okay, so homozygous females and males. So the take-home message here is the more copies of this little duplication, the more extreme the phenotype. So it's probably easier to see it here. So here's the wild type. So here's the bar region. So this red or brown, whatever this is. This is the duplicated region. So here's the normal eye shape. Here we have the duplication. So we actually have 3 copies instead of 2. The eye's taking on a more extreme bar type shape. Here you're homozygous for that duplication. And if you end up with actually even more, let's say 3 copies, you have an even more extreme phenotype. Okay? Yes?
Normally the letter refers to a wheel, but in this case it refers to...
Yeah, it's really talking about this region. Yeah. So this region of the chromosome is getting duplicated, and what you see is the more copies of that region you have, the more extreme the phenotype. Now, does that make intuitive sense? Should it make sense that you have more copies? Why? Why is having more copies not good, or why isn't it better? So what we do is we will see this. This goes back to that whole question about dosage compensation and such. That even in the aneuploids, let's say you have your triploid for chromosome 21 for Down syndrome. It's like, why should that matter? It's just extra copies of the same DNA. It's an imbalance in the amount of the DNA, and cells don't deal well with that. So that's probably what's going on here, is just increased levels of the products for the genes encoded by these regions are what's responsible for this phenotype. Now the question is, how does this happen? There's a couple of different mechanisms, but the most likely and the most common one is something called unequal crossing over. So I've stressed several times in Chapter 2 and also in Chapter 7 that genetic recombination is a reciprocal and equivalent event. So there should be an equivalent exchange of genetic information between the two homologs. It's still not completely known how these chromosomes line up so precisely during meiosis that you get this equivalent recombination, but sometimes they make a mistake. So the chromosomes get a little bit out of alignment. So this should actually be back over here. So now when you get the crossover, you got one copy of the bar region. Then it drops down here, comes over here, and the result is that one chromosome is now deleted and the other chromosome has two copies. So probably the most common mechanism for these duplications is the result of unequal crossing over, simply meaning the chromosomes aren't exactly aligned correctly. And the result will be one chromosome, one homolog will gain DNA and the other one will lose it. Okay. And just sort of give you a couple of examples. These gene duplications are actually quite important in evolution. So you can see the entire gene families. One that's been really well studied are the globin genes. So for instance, beta globin, there's a number of different globin genes in mammals. That one encodes fetal globin, others are adult globin, several other ones. Related to that are myoglobin. It looks like this has been a divergence through evolution where you had this primordial globin gene that then got duplicated. And then those two genes started to diverge a little bit, took on slightly different functions. They got duplicated again, and you end up with a very large gene family. Okay. And I think we'll just to kind of wrap things up a little bit. The reverse of that is just the deletion. This is what we talked about before. That's simply a loss of it. Now, one of the ways that, one question we'll just sort of wrap up is like, how do you do this? How does this happen? It can happen accidentally or it can happen deliberately, depending on what organism you're working with. So, in the early days of Drosophila genetics, you had so-called chromosome mechanics who were making different chromosomes, deletions, and rearrangements because they'd put the flies in basically in an X-ray machine, and the very high-energy X-rays do a very nice job of just shearing right through the DNA and making these nice, clean, blunt cuts through the DNA. Cells don't like that. They start knitting the ends back together, and you end up with these duplications and deletions and inversions and such. So a lot of them were made artificially in the laboratory. It can also occur just randomly from environmental mutagens. Okay, so let's